All right, good afternoon. Happy Monday. Yes, so happy. In fact, this has something to do with sex chromosomes, potentially. You may have seen around campus that there is a feminine product drive going on campus, because we have lots of issues on campus, not just food insecurity and housing insecurity, but also feminine product insecurity in terms of people that aren't able to get those resources. So if you're interested in donating, you'll find those containers around various places on campus. You might have seen that was in the Collegian. But I also thought it would be worthwhile mentioning, since we're talking about sex chromosomes and sex determination, that always, speaking of feminine products, always just, as it says here, decide to axe the female symbol from their sanitary products. Right? So you may be familiar with the idea that the astrological symbol for Venus is the symbol that's also used for feminine products and for females in general. But that doesn't help trans individuals who may identify as male but yet are still genetically female and so still menstruate. And so to be more inclusive, always has decided to get rid of that symbol on their packaging. So the news. Okay, and that doesn't mean all the other tampon and feminine product manufacturers are doing the same thing. I guess we'll have to see if they follow their lead. So back to sex chromosomes, the topic of the week. We may finish it today, maybe a little bit next class, and by next class then we should be moving into the next big topic in class, which is the central dogma, transcription and translation, how DNA and genes encode RNAs that encode proteins. So that's where sort of long-term vision of where we're headed, as soon as we finish with sex chromosomes and a little bit more about statistics and analysis. So here's a quick question for you. We'll ask you to do this on your own or with a neighbor. We'll get back together in about 3 minutes and discuss the answer. But if I told you that there was any chi-square test, it doesn't matter what it is, had a test statistic value of 10.9 and 3 degrees of freedom, based on this table, what's the p-value and what's the interpretation associated with that combination of test statistic and degree of freedom? So, no answers yet. Work on that for a minute or two. Feel free to discuss with the neighbor if you want, and then we'll talk about the answer. So just review. So we have 3 degrees of freedom. We look in that row. We look at where that chi-square test statistic value, sometimes written chi-squared, 10, or approximately 10. So it's in between that range. So how do we get from degrees of freedom to chi-square test statistic value to knowing the p-value? Where's the p-value on the table? These numbers in the table are not p-values. And the reason I'm pointing this out is because when I ask this on homework questions and exams, I often get responses that are chi-squared values, not p-values. The chi-square value is 10. What's the p-value associated with that chi-squared value?
The numbers that are sitting on top of the table.
So you move from that pair of numbers that flank the chi-square value, and then you look up to the header, and those are the p-values. So the chi-square test statistic value with 3 degrees of freedom has a p-value somewhere between 0.05 and 0.01. This bit, the header of those two columns. So in other words, we would write the p-value, the probability is, you can do this in one of two ways, but 0.1 is less than p, which is less than 0.05. In other words, the p-value is somewhere between 0.05 and 0.01. It's larger than 0.01, it's smaller than 0.05. It's in that range. You only have to do this when you use a chi-square table like this. With Google Sheets and with Excel, which if you haven't seen it already, we'll do later today, you can actually calculate the exact p-value, not a range of p-values. So if at any point in the future you have to use a table to do this, you would report a range based on this table. But if you were doing the actual calculation in a spreadsheet, for example, you could just report the one number that is the p-value, which in this case would be somewhere between 0.05 and 0.01. I feel like we should probably do that to make sure that that's actually right. So if you were in Google Sheets or Excel, what did I say the chi-square value was? 10.9. And degrees of freedom was 3. So to calculate P from that, this is written down on the spreadsheet we're working on today, but just in case you want to see it again. Function starts with equals. The function is chi-dist, or the chi-squared distribution, short for chi-squared distribution, parentheses. And then you enter the chi-square value, comma, the number of degrees of freedom, close the parentheses, and that's the exact p-value associated with those two. So it is between 0.01 and 0.05. It's 0.0122 something. So either of these approaches is fine. Use a chi-square table and report the range of p-values that flank the chi-square value you have, or calculate it exactly or almost exactly like this. So most importantly, what's the interpretation now? We've got a p-value. So what does p between 0.01 and 0.05 mean? What's the interpretation? It's either significant or not significant.
So you reject the null hypothesis, so that means it is significant?
There is a significant difference. Yes, you reject the null. Hypothesis, which the easier way of saying that is the second bit, which is the observed and the expected values are significantly different. Significant difference. Bless you. So what's the rule about significant difference? Significance versus not. There's a specific particular p-value that's important in making that decision. That's all there is to this interpret the p-value. You just have to remember one number. 0.05. If the p-value is equal to or less than 0.05, then there's a significant difference between the observed and the expected values you're comparing, which means that if p is greater than 0.05, ns, no significant difference between the two sets of numbers. So when we say ns, which is a common abbreviation in manuscripts and in research literature, ns stands for not significant. What that really means is no significant difference. Not a significant difference. What's another way? That's kind of like a double negative. No difference, which means the same thing. What would you say? If you were trying to say the same thing as it's not different, you might use another phrase. They're the same. Technically speaking, that's not exactly what the statistical test means, but for our purposes, that's what it means. If they're not statistically significantly different, which is p less than or equal to 0.05, then the two data are basically the same. Any questions about any of that? We're going to see the use of the chi-square test several times during the course of the rest of the semester in different contexts, and that's why we started with, a week ago I guess it was now, the poker flip chipping experiment. So I wanted to give time in class, because this is now due tonight, because we've been putting off for a little while working on other things instead. I wanted to give some time in class for you to work on this and to get feedback on it. And I wanted to point out a few things before I release you to do that again for the next probably 10 minutes or so. So first I want to explain a little bit about the headers of this spreadsheet, because we didn't go through this yet. A lot of you have been guessing about what I intended you to put in these columns. Let's walk through it first. And then once we're done with the brief explanation, go make sure that your row is filled out completely. If it turns out that you did not flip your poker chip enough times, which some of you did not, I brought some, so you can do that experiment again if you need. Let me know, and I can pass those out again. And if you've already done this, some of you have, so you've done your whole row, you've done all the analysis and everything already, which is great. Then I've got another task for you to do that's also chi-square based. So pay attention, hold on. We'll be there in just a second. So what we worked on initially was you used Google Sheets or Excel to choose a random number of flips, times to flip this poker chip, and then I asked you to record whatever number that was just by typing that number into that column C. It says record. Just making a copy of whatever number came up in column B. And then you were supposed to flip that number of times a poker chip and record how many bicycles and how many spades. How many heads, how many tails. These have to be entered, some of you did not do this, these have to be entered as numerals like these. Not as tally marks or any other sort of format, not typed. You can't type the word 4 into one of these. It has to be a number so that Excel or Google Sheets can do calculations on these. What's CAT, do you think? It's short for categories. How many different categories of data are we collecting? How many different groups or bins are we putting our observations in? It's heads or tails. It's spades or bicycles. So the reason why is that important for chi-square tests? Why do we need to know the number of categories of data?
Because it's one of the things that's used to math it.
We're going to use that over here when we calculate the number of degrees of freedom. Right? Degrees of freedom is the number of categories there, number 2 minus 1. So that's the why. So everybody should read 2 there, because everybody was flipping two-sided poker chips. You can ignore columns G and H if you want to. That was me being fancy. Those are, if you want to do it, it's a good exercise for using a spreadsheet. I was using that to double check whether or not the column where you wrote the number of coin flips you were supposed to do equals the total number of observations. Right, so here, I was supposed to be flipping this coin once, and I observed 1 bicycle and 0 spades. So these sums have to match. It has to be one observation there, and that was just what I was doing there, was checking that both of those sums match. In other words, this is where you should please check. If you recorded that you're supposed to flip a poker chip 4 times, but you only recorded a total of 3 observations, then you need to come get a poker chip from me and flip 1 more time. Otherwise, the statistical analysis doesn't work. What about expectations? How do you calculate, if you flip the coin, my example here was the random number generator told me I was supposed to flip a poker chip once. So I did. I got whatever I said I got to the left here. So how did I calculate expectations for that? How many heads and how many tails, or how many spades and how many bicycles? Should be half. But I only flipped once, so half of 1 is half and half. The critical thing here, again, for any future chi-square tests you do, is that the number of observations has to total the number of expectations. That's why we don't round. So if you're supposed to flip the coin once and you observed either bicycle or spade, that's 1 observation. You can't have more or fewer expected heads or tails, so we do have to divide it in half and say that you'd expect half of a heads or half of a spades and half of bicycles, which makes no sense at all intuitively. But that's unfortunately how the statistical test works. So if you had, let's see, in what circumstance would you have a case where you'll calculate a fractional number of expectations? Presumably a bunch of us in the room are going to need to do that. Which ones of us are going to need to have those decimal observations? All of us who had what sort of numbers of observations? Odd. Right? Any odd number divided in half is going to not be a whole number, so anybody who had 1 flip, 3 flips, 5 flips, 7 flips, 9 flips is going to have a fractional number there. If you had an odd number or an even number, then it should be a round number. And finally, after you get your observations and your expectations, you can set up the chi-square formula as written there. Observed minus expected squared over expected for spades or bicycles, and then the same clause for calculating the observed and the expected differences for the other coin, for the other side of the coin, spades. Some of you are advanced Google Sheets users, I saw, and know that there's a formula that you can use that you don't have to type in all of those clauses with the parentheses and the carets to do the powers, the squares. What's the name of that formula? For those of you that used it. It's equals chi something. I think it's chi test. If you did that, that's fine. That will give you the same answer as doing this long formula. But I ask you not to do that because there will be times that I will ask you to interpret p-values from a table, and so you need to be able to know how to do this formula because it involves calculating the degrees of freedom. If you use this formula to calculate the p-value, in other words, you don't have to know the number of degrees of freedom or know how to use the chi-square table, but you do need to do that for this class. So this is a shortcut, but it's not a shortcut that's going to help you learn what you need to learn for class. So better if you write out the whole formula. And then after you get chi-square value and the number of degrees of freedom, you can use that chi-dist formula equals chi-dist, and then the chi-square value and the number of degrees of freedom. That will give you the p-value, probability value, in this case 0.317, which is bigger than 0.05, so it's not statistically different. That's the interpretation. So in about 5 minutes, maybe 10 minutes, as usual, I'll circulate, I'll walk around. If you've got any questions or concerns about how to do any of these formulas, how to calculate this, this is a good time to work in person. This will be turned in by Google Classroom. You don't have to turn this sheet in when you're done with your row and you're satisfied with it. Go back to the Google Classroom assignment and just say turn in. You don't have to attach anything, just say you've turned it in. I will check your work here when I do the grading. Finally, for those of you who are already done and are happy with what you've done on this spreadsheet, there's another type of chi-square test you can do with this, because there's another type of data set that's on this spreadsheet. Not just coin flips and whether or not our poker chips are fair or not. What's the other thing we've done that's random that we have a lot of data on this spreadsheet? The whole class, we gathered a new set of presumably random data that we could test. There were two things we did that was random. One was flipping. The other one was figuring out how many times each of you would flip the coin. Right? We asked Google Sheets to make a random number choice from all of us between numbers 1 and 10. So presumably, maybe, this column the recorded number of coin flips, column C, that should have a random distribution of 1s, 2s, 3s, 4s, 5s, 6s, 7s, 8s, 9s, and 10s. But if I scroll down it, for example, I'm looking for 10s. How many times does 10s show up in column C? Once. Twice. So I'm wondering, did this spreadsheet actually generate a random distribution of those numbers? So if you're already done, I would suggest that you practice chi-square analysis by seeing if you can figure out how to analyze those data to see if they're random or not. Okay, so let's get to it. Again, it'll take about 10 minutes or so, depends on needs. Raise your hand if you've got a question about any of the concerns or processes.
Is it?
Yeah, go for it. Back to sex chromosomes. And there will be more practice on this as well, so I'll put together another couple of exercises to work on chi-square analysis if you want practice. So last time we met, talked a bit about sex chromosomes and how 50% of individuals should be, in humans anyway, males are XY, genetically male individuals are XY, and genetically female individuals have 2 X chromosomes. So in humans, XY is male, XX is genetically female. What does that tell us about how genes control our genetic sex? What is it about those sex chromosome constitutions that define who is genetically male and genetically female?
The presence or absence of the Y.
So it's either, there are 2 differences up here. One is presence or absence of a Y chromosome that distinguishes the 2 genetic sexes. What's the other difference between males and females? The ratio or the number of X chromosomes differs between males and females.
This might be off topic, but what about individuals that have like XXY?
Ah, well, that's exactly where we're headed, so thank you. That totally not off topic, totally on topic. So in humans, is it the presence or absence of a Y or the number of X chromosomes that causes sex determination? Presence or absence of the Y. So if that's true, here's a thought experiment. Take half a minute, think about it, then we'll collect answers. What sex would an XO individual be? And what sex would an XXY individual be in humans? What's your prediction? So that is, if you're a human that has normal numbers of all the autosomes, in the case of Turner syndrome, individuals that just have 1 X chromosome and nothing else, zero means nothing, no other chromosome, or individuals that have 2 X's plus a Y. Yeah.
Would they technically be intersex?
A good question. So what are the phenotypes of these individuals? That's what we're asking. So now we're not talking about genetic sex. That's XX, XY. It's a bit more complicated for Turner and Klinefelter syndrome. So XO is called Turner syndrome. And individuals that are XXY have Klinefelter's syndrome. So Turner syndrome individuals, for the most part, look phenotypically female, but are usually sterile. And there are some other differences in morphology as well. What about Klinefelter's, XXY? If the Y is what does male-female genetic sex determination, then an XXY individual would be,
Typically female?
they've got a, so there's 2 X chromosomes. This is why this sex chromosome constitution, why people with Klinefelter syndrome are really useful just in our understanding of how genes control sexual development. They've got 2 X chromosomes, like females, but they've also got a Y chromosome, like males. So this is the really important question. What do they phenotypically look like? That is their morphology. Do they look more like a morphological female or morphological male? What do you think? Who thinks it's closer to female? Raise your hand. Closer to male? It's closer to male. Of course, every person who has Klinefelter syndrome or Turner syndrome is a different person, and there's a spectrum of different phenotypes, traits, the way they look. But in general, Klinefelter syndrome individuals look more male than female, perhaps, but are also usually sterile. So in humans, the take-home message here is in humans, it's the Y chromosome that does sex determination. If you have a Y chromosome, then that produces a male most of the time. And two X chromosomes, a lack of a Y chromosome usually means female development. There are a bunch of caveats that we're going to get to in a minute about this. But first, let's contrast this with what happens in Drosophila melanogaster, the fruit fly. Same categories of genotypes, XY, XO, XX, and XXY. XY Drosophila fruit flies are male, just like humans. XX Drosophila are female, just like humans. So here's the trick question. What's an XO fruit fly? Male or female? Male. And an XXY fruit fly then must be female. What the hell's going on? It's because humans and fruit flies, despite having X chromosomes and Y chromosomes, do sex determination totally differently. In humans, as I said, it absolutely is the presence or absence of the Y chromosome that causes male development. So what's different about fruit flies? It has nothing to do with the Y chromosome.
The chemicals that are coded by the sex chromosomes?
It is triggered by the sex chromosomes, but not the Y. So what's the other genetic difference between males and females? Presence or absence of the Y, or the number of X chromosomes. So in Drosophila, it's the number of X chromosomes that dictates sex development, not the number of, or presence or absence of the Y. In other words, the XY fruit flies have one X chromosome. The XO fruit flies have one X chromosome. They both develop as males. The XX and XXY fruit flies, and these have been created experimentally to know what these fruit flies look like. The XX and XXY fruit flies both have two copies of the X chromosome, and they develop as females. So the goal with this exercise is not only to point out that genetics is pretty varied and pretty interesting, but also that you need to know there are two different ways that sex determination can occur when there are two sex chromosomes. It can be it's the number or the ratio of one of the sex chromosomes, like the Y, or the other one. So in other words, in Drosophila, the males have Y chromosomes, but the Y chromosome has nothing to do with sex. It's just it's along for the ride, basically. Any questions about that? There are lots of ways that organisms use genes to determine sex, but these are sort of the big two categories. Presence or absence of one sex chromosome, or the number of the other sex chromosome. So getting back to humans then, genetic males are chromosomally XY and females are XX. That's basically a binary decision. You can't have any other than XO and XXY. The presence or absence of the Y is binary. You either have a Y chromosome or you don't. It's kind of oversimplified. So we're going to get into some details in just a minute. But it's a binary thing. You either have a Y or you don't. You either develop as a genetic male or you have the genotype of a female, XY. How is this not true for phenotype, for our traits, for our behaviors, for how we look, how we act? That is an XY male phenotypically male?
No.
Not necessarily. Why not? Yeah, there's transgendered individuals.
Sometimes people just develop more rounded faces, or as in females, just smaller chests, or...
Right, this is how do you, this gets into a really important question about how do you define phenotypically male and female. We've talked about genetically how we define males and females. I can collect DNA, I can't, I'm not allowed to. I don't have permission yet. I could collect everybody in theory. I could collect everybody's DNA and get a karyogram for everybody and know if you have 2 X chromosomes or an X and a Y. Or an X and that's it, Turner syndrome or XXY. You could do that, and you can use that as a basis for defining genetically what sex people are. Phenotypic is way harder. There's no one-to-one correlation between sex chromosome constitution and phenotype. Because phenotype, our traits are developed not just because of genetics, but also because of the environment, nature and nurture. And that's not to say, and this is really uncomfortable for a lot of geneticists, because scientists tend to like to be sort of objective people that have numbers and make decisions, and we like to have rules. We like to know what the rules are, rather. We like to know that nature follows the rules. But that's not at all the case some of the time in genetics, where it's not just a simple, do you have a Y chromosome, then you're male. Because there are lots of other genes that control development and the development of the body, aside from the Y chromosome saying you're going to be this sex. And so it was pointed out, they're intersexual individuals. Something like 1% of human births, the immediate phenotypic sex of the baby is not clear. They're somewhere in the continuum of phenotypic sex. That is, they might have a penis, but it looks like the start of the development of a vagina, a hermaphrodite. Something like 1% of live human births are not, it's not clear. They have some parts of both types of genitalia. And often it's the case, or at least it has been in the past, that at the point of birth, if the doctor noticed that, they would talk to the parents and they'd say, what sex is your baby? Like, what sex do you want them to be? Sometimes there was surgical intervention to fix the problem if it was a problem. Like if it was mostly boy and a little bit girl, then maybe there would be a surgery to remove the rest of the girl-like phenotype to make it clear. This is in the past. This probably still happens today. I'm not aware of it, but at least not in this country, but it probably does happen in this country. So phenotypic is different, and that's because the sex chromosomes are not the only components of sexual development. They're not the only thing that drives how we look, how we act, whether or not I've got an Adam's apple, the size of my Adam's apple, right? It's supposed to be a male-only trait, but it's present to small degrees in some women too, and vice versa. So, in other words, the connection between genotype and phenotype is not clear. Genotype is binary. You have a Y chromosome or you don't, but then the way you look and the way you develop as a child and then as an adult is varied. There's a continuum. That said, though, it is important to know, let's see, we could have a really long discussion, and maybe we should hold it off for next time, but I'll raise the question anyway. Is there such thing as a sort of pure male? That is are there individuals on the planet that are, without any doubt, completely male, if that's a thing, or completely female? Or are we all in this continuum between mostly male and mostly female? I don't know. Think about it. But one of the things that we do have to understand is that there are hermaphrodites, individuals that have some part of both development of both sexes in the same body. To my knowledge, though, no humans are hermaphrodites to the extent that they actually produce functional eggs and functional sperm in the same body. There are some vertebrates that do, not humans, but there are some vertebrate species that actually can, in the same body, produce sperm and eggs in the same individual. How do we determine, by looking at a person, how would a biologist, you're a biologist, how would a biologist define, if you went out into the field for the first time, you discovered a new species, and you said, I wonder what sex this individual is. How do you know? What's the biological definition, not genetic definition, what's the biological definition of sex? Right. So in biology, the females are the one, as we discussed, right, that produce the large immobile gamete, the oocyte. we call it an oocyte in humans, but other species produce the large immobile gametes. That we don't call them eggs, we call them something else. So phenotypically, that's sort of the biologist's general definition of the female. Whichever individual produces large immobile gametes, and the males produce the small, sort of disposable, is a good way to put it. Dispensable, maybe, disposable. And numerous, and usually mobile gametes. Those are the males, produce those sorts of gametes. That's the biological definition of sex. And there are individuals that can do both at the same time. Not functionally, but those are, among other people, hermaphrodites. This doesn't work for all species, though. This concept is called anisogamy. I think I mentioned this. Did I mention this already? Yeah. Anisogamy. The two gametes are different sizes. Does anybody know of a species that has isogamy, where two individuals produce gametes that are the same size? So we don't call them male and female because we can't tell the gametes apart? They exist. Yeast are one of them. So baker's yeast, Saccharomyces cerevisiae, or brewer's yeast, depending on which you prefer, bread or beer. They produce gametes that are the same size. So we don't call them male or female. In yeast, we call them A and alpha. So one sex is A, one sex is alpha. Right? We don't call male and female, we just use a different set of terms. It's still a binary thing, though. It's one or the other in yeast. Sometimes both, maybe. So, summary, sex chromosome is a binary thing. You either have it or you don't, but that doesn't necessarily cleanly translate to phenotype. True of most things in biology, or in genetics. There are very few traits where the presence or absence of a gene gives you that trait. You either have it or you don't. There are a lot of traits, diseases, and height, IQ. Oh, speaking of genetics in the news, here's a rather unfortunate case that I just saw today while I was waiting for Biology 104 students to come do some of their experiments in molecular and cell biology lab and genetics. Parents who are were divorced, are battling over the custody of their child, and the parents can't agree on the sex of the child. So this is a hugely important case because what do you do? How do you award custody of a child to two parents that want to raise the child as two different, like intersexual versus one sex? Thanks. And then another study recently. Fortunately, and you may have heard about this, designer babies. We talked about the ability for humans now to get the first polar body and the second polar body from oocytes and predict the chromosomes that are in the oocyte for in vitro fertilization, make sure that's a good oocyte to use, good in quotes. But there are these, because geneticists know so much about how different genes interact with different traits like intelligence or height or eye color or hair color or whatever, now there are companies that are trying to develop these screening technologies. So this is not an oocyte anymore. This is an early embryo. So it's an in vitro fertilized oocyte that started to divide and produce an embryo. And early in development, really early in development, you could suck off a couple of those cells and it doesn't harm the embryo. Embryos are plastic enough in early development that you can get rid of some cells and they'll just create an otherwise normal human anyway. So you could get some of those cells and test them for the presence or absence of these genes, like genes that maybe control IQ. You collect your many in vitro fertilized embryos for the ones that maybe have the best genetic quality according to you. Fortunately, and this is just to reinforce the point was just being made about sexes and the fact that there's a continuum, that there's nature and nurture in intelligence, all of these other traits as well. So it's very unlikely, even if you could, and even if you had the money, that you would accurately be able to predict the IQ, height, stature, weight, strength, speed of any human just based on genetic tests. And in fact, so this study was done, and they actually point out that people that want to do this might accidentally throw out the embryos that would be their tallest or fastest or whatever kids. Because the genetic testing, we don't know about the nature component of genetics yet. Okay, last thing before we go. There's the score distribution from exam 2. Ranges from 100% to 1. Nobody got 0. And a pretty reasonable distribution. So here's what I'm asking. Tomorrow, because this is a critical point in the term, this is where things either get better or get worse, I want everybody in the class, I want everybody in the class to get better. But this is the point when people tend to either decide to tune in or tune out. So tomorrow, I've canceled all of my appointments, meetings, and everything else. I'm going to be in my office all day. If you want to come talk to me about anything in class, anything, please do. I'll be in my office all day, Science 326. Especially if you're in the 7 or below range, I really expect you to come talk to me for 5 minutes. If that, just come say hello. Let's visit briefly, or for a long time if you want. And I'm going to be sending out the total scores for everybody and posting them on Canvas at some point later today when I get time. So everything's scored. And the key is already online. The video key is already on YouTube. So you can check your answers against the key, see how you did. And next time we come to class, we've got those exercises, 7.6, 8.2, and 8.4. We're going to keep rolling.